So sensory neuron is on top, motor neuron is at the bottom. On the left-hand side, you see the tail activating serotonergic cells. Let's look at the basal condition. In the absence of tail stimulation, we generate an action potential in the sensory neurons. As Tom Jessel pointed out in his lecture, that propagates to the presynaptic terminal. It opens up calcium channels, allows calcium to come into the presynaptic terminal, and calcium is absolutely required for the exocytotic release of transmitter. It allows vesicles to bind and to be released. What happens if we now stimulate the tail? We activate serotonergic cells. These release serotonin. Serotonin is recognized by a serotonergic receptor in the presynaptic sensory neurons. These engage an enzyme called an adenyl cyclase that converts ATP to cyclic AMP. Cyclic AMP is a second messenger. There are lots of different second messengers. They all have the same function. They carry information from the membrane surface to the molecular machinery inside the cell. Cyclic AMP activates a particular molecular machine called the cyclic AMP-dependent protein kinase. That is abbreviated protein kinase A, pKa. Cyclic AMP-dependent protein kinase has two subunits, a regulatory subunit and a catalytic subunit. I've indicated the regulatory subunit as a spindle-shaped structure and the catalytic subunit as an oval-shaped structure. The regulatory subunit inhibits the catalytic subunit under normal circumstances. When the level of cyclic AMP rises, the regulatory subunit binds cyclic AMP, undergoes a conformational change, frees the catalytic subunit. It now can move into the presynaptic terminal. It acts on ion channels. It phosphorylates them, alters their properties, allows calcium to come into the presynaptic terminal. So now an action potential has more calcium coming in, more vesicles can bind, more vesicles can be released, and you have enhanced transmitter release. And that is the mechanism of short-term memory for synaptic facilitation. And this is occurring at every single sensory neuron that is involved in the reflex.